Hello, welcome to Teaching Bio. In this video, we're going to look at the next part of the controllable water potential, and this is the loop of Henle, which is the most confusing part. So the most important thing to remember with this are the statements on the slide currently, and that is that the descending limb is permeable to water, but impermeable to ions, and the ascending limb is permeable to ions, but impermeable to water. Now, it's pivotal that you remember these two sets of statements, and that will then make everything so much easier to understand. So, as I said before, there's this golden rule that water follows ions. Well, yes, that is the case, but here it actually isn't the case, which is what makes this confusing. And that's down to the differing permeabilities, but we'll look at that. So, we're going to start off nice and simple at the descending limb. So getting our bearings with this picture, this is the descending limb, and this is the ascending limb. This is the medulla, which is a type of tissue found in the interstitium, which is all the bit that's not the loop of Henle. And there's this increasing hypertonic interstitium, i.e. it's losing water as you go down and it's getting saltier, which we'll look at. So this region will be salty, this will be saltier, and this base region will be the saltiest. So, following on from before, so here is our filtrate from the proximal convoluted tubule, and it's moving along down the descending limb of the loop of Henle. And as it goes down, it starts losing water because there's a high water potential here and a low water potential here. So water starts leaving, as it does, moving down the water potential gradient by osmosis. So it leaves the filtrate passively via osmosis. Now, because water's leaving, the filtrate starts becoming more and more concentrated, which logically makes sense. So now it's much more concentrated. And as we get to this point here, it's very, very concentrated. Remember that it's got ions in here, and normally the ions will sort of try and follow the water, or vice versa even, but they can't because the descending limb is impermeable to ions. The ions are remaining there, which is why it's super salty, because there's tons and tons of sodium and chloride ions left there, and hardly any water because all the water's just been pumped out. So the filtrated water potential decreases, it descends because the water's being lost and it's jam-packed with lots of ions that can't follow. Okay, now we're going to move on to the ascending limb, and then we're going to look at the thin ascending limb at first, so we understand this part. So that really, really concentrated filtrate now starts to lose some of its sodium. So some sodium is reabsorbed in the thin ascending limb via facilitated diffusion, because the thin ascending limb is permeable to ions but impermeable to water. So sodium can leave, and it leaves by facilitated diffusion from a higher concentration to a lower concentration. So sodium starts leaving. And as sodium starts leaving, the filtrate becomes less concentrated, and even less concentrated. However, the water that's in the filtrate cannot follow the sodium. So the interstitium starts to become more salty, because obviously all this sodium is leaving, so it's salty. The water can't follow, so the water starts becoming more dilute. Nice and simple. So the sodium is being lost by facilitated diffusion, but the water can't follow because the ascending limb is impermeable to water. The water is remaining in the ascending limb. Therefore, the filtrate is becoming more dilute. Now we're going to look at the thick ascending limb. So as we go along, let's see what happens. Okay, so the filtrate's moving along, and this time, even more sodium is going to move out, and it's going to take some chloride with it using the sodium chloride transporter. So now we're going to pump sodium into the interstitium using the sodium chloride transporter, which is going to function against the concentration gradient. So now we're moving from an area of low sodium concentration in the ascending limb to a high sodium concentration, which makes sense because we had tons of the sodium here, but we pumped out some of it at the base. So that could move by facilitated diffusion without ATP because it's not going against a gradient here. But at this point, there's a lot more salt there now, so it has to use ATP. So we're going to pump out the sodium against the gradient. And as the sodium leaves, the water can't follow it because remember the ascending limb is impermeable to water. So the water starts becoming dilute. And we move along, and now it's very dilute. And we have produced a hypertonic filtrate, which is a very high water potential relative to this bit here at the start and this bit here at the base. So the point of this is to produce filtrate with a high water potential. Okay? So all nice and simple concepts. So where do these water ions go though? So we keep saying that the water is pumping out and moving in and doing all of that stuff. Well, the water is being pumped out into sort of the vasorector. So you don't have to get worried about this, but this is just what it's called. It just gets moved down to there, and so does the salt. So this is some text, just explaining everything that I went through. So you can pause this, read through it, and have a go. And then there's some more text here, so you can pause this, read through this, and write down your notes or whatever. Okay, so it's very simple concepts. The key thing to remember with this is that the ascending limb is impermeable to water, but permeable to the solutes, and the descending limb is permeable to water, but impermeable to the solutes. So some key points from this, which they might use as an application point in exams, is that the longer the loop of Henle, the more concentrated the urine is, because we've got more time to reabsorb water down the length of the descending limb, which remember is permeable to water, and that was where the water was being pumped out initially. The entire limb is actually impermeable to urea, and that will remain within the filtrate. And then that will then be passed out into the urine, or we will reabsorb at the collecting duct, which we will look at in a minute. And remember that half the urea we reabsorb in the proximal convoluted tubule before. So, moving on to the collecting duct. The collecting duct is actually quite permeable to water. And because of the way in which the loop of Henle works, in that it produced a filtrate that had a very high water potential, we can control the movement of water via ADH. So any water in the medulla has already been absorbed, and so the water potential is higher in the collecting duct, therefore we can move water out of the collecting duct. So we'll look at that in a minute. So remember that the ascending limb of the loop of Henle, the distilled convoluted tubule, and the collecting duct is impermeable to water. So water reabsorption can occur in the late DCT and collecting duct in the presence of antidiuretic hormone, ADH. And the way this works is it promotes the fusion of aquaporin channels, and more specifically AQP2 channels, into the cells of the late DCT and the collecting duct. Therefore, we can reabsorb water by osmosis into this interstitium, and that is driven by that gradient that we produced via the loop of Henle, therefore producing concentrated urine. So again, this is the loop of Henle. Remember, we had a gradient here where we had very, very salty, saltier. We had salty, saltier, and saltiest. So because of that gradient, we can continuously move water out of the collecting duct. So there's our gradient, and this is the movement of water out of the collecting duct and the DCT into the blood, into the vasorector, into those cells, producing concentrated urine. And water will always move out down the water potential gradient. So water potential will be higher here and lower here, as you can see. It's higher there and lower there, higher here and lower here. These values here represent sort of like the concentration of the ions. The body can therefore control how much water we release into the urine by regulating how much we actually reabsorb. So if we reabsorb buckets of water here, then we're going to produce a very concentrated urine. And then a very dilute urine will not have as much water leaving, as all the water will remain there. And then that is controlled depending on ADH, which is released from the posterior pituitary gland in the hypothalamus. And therefore the balance of the blood water potential depends on this. So if we have lots of water in the blood, we're going to expel that in urine and have less reabsorption of water, and then vice versa. So this is how ADH works. It will get released from the posterior pituitary gland in the hypothalamus, travel in the bloodstream to the kidneys, and bind to ADH receptors. Inside the cells, we've got these watery vesicles with aquaporin channels. So when it binds, it triggers a series of reactions and activates an enzyme called phosphorylase. And then while it does it, it sort of moves these vesicles to this membrane here of the epithelial cell, lined in the collecting duct. These can then be embedded into that. Therefore, the water that was in the lumen, that filtrate that we just produced, can then move in to the cell and then into the blood. So, as I said, ADH is released from the posterior pituitary gland and hypothalamus moves to the kidneys and binds to those receptors of the late DCT and collecting duct. This then leads to a series of enzyme-controlled reactions and produces and activates the enzyme phosphorylase. And then inside the cell, as I said, there were these watery vesicles containing aquaporin channels and more specifically AQP2 channels. Phosphorylase will cause those vesicles to fuse with the membrane of those cause those vesicles to fuse with the membrane from those cells, and therefore release the aquaporin cells there by exocytosis, so that water can be drawn out of the collecting duct into the cell via AQP2 and then into the blood. And then ADH does a similar thing with urea, because I remember I said before some of the urea will be lost in the urine, so will be reabsorbed. And that will stimulate more sort of enzymes to make specific urea transporters, and then we can use the exact same mechanism here to draw the urea out of the collecting duct into the cell and then into the blood. And then reabsorption of water is a response to low water potential in the body, and that is the most fundamental statement to take out of this. So this here is a slide summarizing the responses to having too much water in the body and too little in the body. When we have too much water in the body, we want to expel that water, so we want to wee more. The reason why we might have too much water in the body is because we've drank a lot of water or we have very little salt. And as a result of that, we have a high water potential of the blood. Therefore, the osmoreceptors will detect this high water potential and they will release less ADH. So by releasing less ADH, we get less of the aquaporin channels. Therefore, we will get less water reabsorption from the collecting ducts into the cells and into the blood. So more water will be expelled into urine, producing very dilute urine and therefore lowering the water potential of the blood. And the same story is true, it's a different story obviously is true vice versa, where if there's too little in the body because we haven't taken enough water, we have too much salt, we'll have less water potential, the osmoreceptors will detect that in the hypothalamus, will release more ADH, therefore we'll get tons more aquaporin channels, so we'll get more and more water being drawn out of the collecting duct into the cells and into the blood. So this slide here effectively summarizes what happens to the products as we go along the nephron. It's important that you have this picture in your mind because this is what they're likely to assess in different application scenarios in the exam. So make sure you know that all the glucose will be reabsorbed at the glomerular, will be pushed into the lumen of the Bowman's capsule, therefore it will be in there, and then it will be reabsorbed at the proximal convoluted tubule. So you should not have any glucose here. And in diabetics, they do have glucose in their urine because of sort of damages to blood pressure and changes and lack of permeability. Increase in permeability, meaning that more glucose get forced in. Similar story with amino acids. And then again, proteins, you shouldn't have proteins in urine. If you do, there's some form of damage. Where will the damage be? Well, if you've got large proteins, the likelihood is the damage is at the level of the glomerular filtrate, because that should act as this filter, this sieve stopping the proteins moving in. And then the water, obviously there'll be water along the length. You will have water in your urine, and that will depend on how much obviously is needed to be reabsorbed and how much needs to be expelled. We will have urea along, again, the length, and we'll have salt along the length, and then there'll be some left. It will fluctuate because a lot will be reabsorbed, but we will still have salt in our urine because urine is indeed salty. Okay, thank you very much for watching, and please subscribe.